name is Sasha Jenkins. I'm a soil scientist and a microbiologist at the University of Western Australia. I'm part of the NAM project, and this part of the project is looking at developing the best management strategies in terms of reducing greenhouse gas yield, but at the same time trying to improve crop productivity and soil quality. First of all, we essentially screened all the manure types across the whole of the intensive livestock industries, and we identified the lowest emitters. They were then taken forward to a next round where we found the best way to apply them. Then we did a next experiment in the lab, was to then identify who's responsible for the greenhouse gas in terms of the microbial populations, and in knowing that, we then modified the farming practice again so we could again lower the greenhouse gas. We then, taking all that information together from the first 3 laboratory trials, we put that information together to design a field trial to capture how to lower the greenhouse gas. Basically what we found was the pelleted manure, the composted manure and the stockpiled for 12 months were the best in terms of lowest emitters. But it's better to put it at 5 centimetres and incorporate it. And the main key bugs responsible for producing greenhouse gas in this field was nitrifying bacteria, and we basically, to avoid when those bacteria were most active, we basically dry seeded to keep it sort of cool, environment cool, and to avoid the rainfall events, which basically stimulate them. And we also placed them at the manures at a sort of 5, because the populations were highest at 5 centimetres, so to avoid them, we basically put the manures away from them, essentially it. In this field trial, we're looking at wheat, and each one of these strips, I think maybe you can see them from the pink flags here, each one of these strips actually represents a different treatment. So these treatments are in random block design, so randomly distributed through the soil, through the actual plot. Each treatment's replicated 3 times, and I think we can really start to see some of the difference emerging between the different treatments. This is the fresh plant material we collected in the field. It's then dried down, and we then weigh it to work out its dry mass. This gives us an indication of biomass, or an indicator of how well the plant's growing. We then also measure the nitrogen content of the plant, and this gives us an indication of how much the fertiliser or manure is delivering nitrogen to the plant and how much is escaping as nitrous oxide to the atmosphere. When we come to the field trial, obviously I think you appreciate we can't run every single manure treatment in a field trial because it has to be replicated, and it's also on 2 sites. We have a clayey soil and a sandy soil. So the first stage was really just to get a baseline of what emissions were coming out of those by applying those manure types to soil in terms of both a sandy and a clayey soil. And this is really important because at the time Australia did not have a baseline study for baseline information on greenhouse gas emissions for its industry, and we were actually using information from Europe, and I'm sure you can appreciate that the systems in Europe, the soils are completely different. Even the way we house their livestock and feed their livestock is completely different. So we don't really know if the values or the numbers that come out of Europe can actually be transferred to Australia agriculture. So it's very important to get a baseline of all the manure types so we can identify which are the best ones to take forward into subsequent trials.
Hello, my name's Ian. I help run the field trial here, and this is the gas chambers that we use. So what we need to do first is we need to turn the fans on. We then place the chamber lid on, put the bungee cords onto plastic toggles. We're now going to take 20 mils for our gas sample from the internal chamber. And push that into the chamber. Remove. We collect our gas samples over a 1 hour period at 0 minutes, 20 minutes, 40 minutes and 60 minutes. The vial is then taken back to the lab and is measured for 3 gases, which are methane, CO2 and nitrous oxide.
So the second series of experiments, this was really to identify strategies in storage, which was the best way to store the manure before it was being applied to the soil. So essentially with our effluent, it was whether we put it in a covered pond or whether we don't put it in a covered pond. With the more solid manures, we were looking at fresh manure, we were looking at various stages of stockpiling from 3 months, 6 months, 12 months. We're also looking at semi-composting, conditioning, type composting through to proper composting, composting with extra additions of carbon inputs. Some of the industries are also looking at pelletizing that, and the main reason we want to pelletize is it's a lot easier for the farmers, the grain growers particularly, to put that down with their air seeders. So it just means that it opens up that market to grain growers. The benefits or the ramifications of this project is that it's going to potentially benefit two industries. One, the intensive livestock industries that produce the manures, the compost, the pellets, because they're going to actually be able to finally convert waste to dollars. And the second is the grain growers that are going to potentially utilise that product in their land to see improved crop productivity and also other soil benefits. So it means that their costs, their input costs are going to be greatly reduced by potentially using this product, and they're going to see the benefits in soil structure and soil carbon, water holding capacity and other benefits from adding manure to the soil. So they're really going to profit from that, and hopefully this will encourage the growth of both those industries.